What if I told you that this pig heart right here could one day save a human life? This could be the key to solving one of the greatest medical challenges of our time, the demand for human organs for transplantation exceeding the supply. In 2022, a pig heart was successfully transplanted into a human patient. It functioned not for minutes, not for hours, but for 7 whole weeks. The question is, how did we get here? Today, we're exploring the major immunological barriers to xenotransplantation, that's transplanting animal organs into humans, with an in depth look at Sykes and Sash, covering genetic engineering, immunosuppression, and tolerance approaches to make this thing survive in humans and start a new era for organ transplantation. In the US, every day 20 patients die while waiting for organ transplants, while a new patient is added to the waitlist every 10 minutes. There's an organ shortage with over 116,000 people on waitlists, and since only 1% of deaths result in viable transplant organs, no amount of human donations will ever meet the current demand. Alternative strategies like bioengineered organs through methods like 3D bioprinting lack detailed mappings of cells within an organ, making replication of the microenvironmental niche extremely difficult, while mechanical devices like perfusion pumps are extremely costly, making them prohibitive for most transplant centers. That's why xenotransplantation, particularly with pigs, offers some hope. Why pigs? Well, pig organs are anatomically similar to human organs, and pigs grow to different sizes, producing organs for different patients. Pigs also breed rapidly, with large litters that are readily available. We slaughter around 3.8 million pigs every day for human consumption, so there aren't any ethical objections to using their organs, and they are suitable for genetic engineering. The issue is we can't swap human organs with pig organs without overcoming immune barriers. To add real-world context, I emailed Dr. Mohammed Wahid, a pioneer in xenotransplantation and the surgeon behind the very first successful pig-to-human heart transplant at the University of Maryland Medical Center back in 2022 that is cited in the article. And guess what? He responded to my interview questions in writing. I'll incorporate his comments to see how the Sykes and Sash translates to clinical practice. The human immune system is trained to recognize foreign and non-human tissue. Sykes and Sash details that rejection of pig organs happens because of a cascade of immune responses leading to hyperacute rejection, an attack on the transplanted organ within minutes to hours. Looking at the xenotransplantation milestones, in the 1980s, the survival of pig organs was minutes. Today, pig islet cells and kidneys last years in non-human primates. What changed? Well, CRISPR-Cas9 and transgenics allow them to modify the pigs in several different ways. Essentially, when a pig organ is transplanted, it is immediately attacked by pre-existing antibodies that see it as foreign. The antibodies bind to the pig-specific molecules like GAL and trigger a complement cascade where proteins activate each other to burst bacteria, cause inflammation, and mark the foreign material so other immune cells can find them. To avoid this, the pigs are genetically modified to knock out the GGTA1 gene, which removes the GAL antigen. But that's not enough. Humans naturally make antibodies against certain antigens that exist on the pig cells but not on human cells. The two key antigens are NEU5DC, made by the CMAH gene, and CDA, made by the B4GAL-NT2 gene. So the pigs are further engineered to knock out CMAH and B4GAL-TN2 genes, creating triple knockout TKO pigs that reduce human antibody binding. Regardless, the complement system proteins that kill invaders and attract immune cells will still be activated. Pig cells can't regulate the human complement efficiently, so complement proteins will bind to the pig cells and poke holes with the membrane attack complex to cause cell lysis and inflammation. So the pigs are then engineered to express human complement regulatory proteins CD46, 55, and 59. But the issue is that complement activation, part of your innate immunity, can still activate clotting factors, leading to thrombotic microangiopathy or TMA. These small blood clots that form in the pig graft vessels, cutting off blood supply and killing the transplant. To solve this, the pigs are modified to express human anti-clotting proteins, human thrombomodulin or HTBM, and endothelial protein C receptor. Our innate immune system further complicates this because macrophages attack cells that don't have the CD47 signal, and natural killer cells will destroy cells without human MHC1. So the pigs are engineered to express human CD47 or the don't eat me signal, and HLE, a type of human MHC1, and beta 2M, a component of MHC1 genes. The adaptive immune system comes in to use T cells to attack the transplant. T cells attack pig organs in two ways. Direct recognition, where T cells will see the pig MHC and attack the pig organ directly, and indirect recognition, where human antigen presenting cells, or APCs, will digest pig proteins, present them on the human MHC, and cause T cells to trigger a strong immune response. This causes a cytokine storm, an immune activation that can damage the graft and the patient. Even when immunosuppressants are used, this T cell response is hard to control. So what we do is we train the immune system not to attack the pig cells with two strategies seen in Figure 2. Here you can see how tolerance was induced across the pig primate barrier, and the article suggests that the same can be done with humans. So let's look at Figure 3. First up, thymic transplantation. The thymus is an organ that contains special cells called the cortical thymic epithelial cells, which are responsible for training immune cells like T cells to recognize what belongs in the body and what is foreign. By transplanting the thymus, we're training the immune system to accept a transplanted organ. Essentially, this figure shows that a hybrid thymus, one that was injected with human CTECs, taken from the organ recipient, trains T cells to recognize both human and pig markers, allowing immune tolerance of the transplant and making sure no infection occurs. The other strategy is mixed hematopoietic chimerism. When we look at Figure 2 again, they transplant pig bone marrow into the thymus of a recipient baboon and create a mixed immune system that over time will learn to tolerate pig cells because the T cells, B cells, and NK cells are re-educated. What Figure 3 shows is that combining thymic transplantation with mixed chimerism, where both recipient and pig immune cells are in the body, further prevents rejection. According to Sykes and Sash, the best setup is a hybrid thymus with mixed chimerism to ensure the transplant is accepted and still working properly. These methods have successfully tolerized the T cells, B cells, and NK cells of genetically modified mice when human immune systems. It's working, considering Dr. Mohib was able to transplant a human patient with a pig heart with 10 genetic modifications that worked for 7 weeks. But when asked about key immunological hurdles that need to be fixed before making xenotransplantation acceptable, here's what he wrote. Antibody-mediated rejection and the condition of the patient that opt in for xenotransplantation has been a real issue with us. We have learned a lot with the 2 transplants and now know what to avoid, like IVG, that may have induced antibody-mediated rejection in our first patient, and blood products, which is the same in the second one. The patients will have to be managed carefully and will have to deal with the problems as they come along. While considering infection risks, particularly porcine endogenous retroviruses, or PERVs, embedded in the pig DNA, Sykes and Sash reported none in the xenotransplant cases. Dr. Mohidin reassures we and others who have done xenotransplantation in humans have not seen PERV activation. One company has made pigs with 59 PERV genes knocked out. We plan to monitor patients constantly. While xenotransplantation promises a solution to the organ shortage crisis, let's break down the EDII lens. If xenotransplantation becomes a reality, will these organs be available to all patients or just those who can afford the procedure? Well, the article doesn't consider it, but on average, countries in North America and Europe, who are the leaders in organ donation, have 4.1 times more physicians, 9.8 more times surgical workforce, and 3.5 more beds, ICU beds, and transplant centers than countries and regions such as Southeast Asia. So are the wealthiest the only ones who can afford a second chance at life? We also need to consider how different communities view xenotransplantation. Certain indigenous, religious, and racialized groups have a historical distrust with experimental medical procedures with a legacy of medical racism. Some religions have strict rules when using animals in medicine, but also in their cultural practices. And while you can argue that pigs are farm animals and don't require ethical consideration, we need informed consent from the involved patients. According to Dr. Mohidin, we are rapidly resolving all ethical concerns. A major concern is that a patient who doesn't have any other option will consent to xenotransplantation in desperation. This is not the case, as we take 4 psychiatric opinions for our first patient just to see if he understands the process. We design lengthy consent forms to make sure we cover all possible issues that can happen. The biggest challenge? Funding. We need more support from government and industry to move the field forward. So what's next? We know that pig organs have survived 9 months in baboons, years in NHP kidneys, and 7 weeks in humans. Well, my interview question, looking ahead, how do you think clinical trials for xenotransplantation will evolve in the next few years? Well, Dr. Mohammed said, I think we will learn a lot from clinical trials. The success will come in increments, and we will get to a stage where we can offer this procedure to all patients. So if you needed a transplant and a pig organ was your only chance, would you take it?